Okay. Yes. Yes. इसको minimize करना। इसको minimize करना। ऐसे तो speaker जो भी होगा। नहीं पहले आप कुछ बोलेंगे नहीं। ये बोलेंगे तो कैसे?
A very good afternoon to everyone present here today. I extend a warm welcome to each one of you on this magnificent afternoon of celebration. Today, I feel privileged and honored to stand here as we celebrate the immense power and strength of all the incredible women around us. International Women's Day is celebrated across the world on March 8th to recognize and empower women. Empowering women is not just a goal, it is a necessity. This year's theme for International Women's Day is to embrace equity. It calls for us to realize that equity isn't just a nice to have, it is a must have. Equality is leaving the door open for anyone to approach it. Equity is ensuring that there is a pathway to that door for those who need it. To push for excellence today without continuing to push for access means very little. So be incredibly and ruthlessly selfish with your equity. They'll tell you that you are too loud, that you need to wait your turn and ask the right people for permission. Do it anyway. Remember that well-behaved women really make history. A very happy International Women's Day to every one of you. With this, I now request our director, sir, to come up to the podium and deliver the welcome address.
So thank you for starting off today's program. As you have rightly pointed out, it is a day when you are celebrating a very important part of our everyday life, which is celebrating the power of women. But this should not be only for one day. It should be on every day of our life where we should celebrate the same occasion. Because ultimately this society is built because of men and women joining hands together. As I stand here, I want to emphasize again that this celebration should not be restricted to only one day of a particular year. It should be done every day. And by that, I mean the celebrating and empowering women in every aspect of our life. This brings me to a situation which I would like to talk to all my colleagues and also my student friends. It is actually very sad that as we go up the strata of life, because of prevailing social situations, the participation of women in different fora becomes less in our country. And that is something, it's a question of sadness, and also it is a question of introspection, why it should happen. While I understand there are societal pulls and pressures on women and unjustly on women, because many times their careers are kept on hold so that the male counterparts can go ahead. This is something which every person in the audience today, we have to make sure that it is not only me who has to go ahead, it is the equal participant in the life who have every right to go ahead. And that is something the younger friends of mine who are in the audience should always remember all through their lives. it is not only for one day we are celebrating the Women's Day. We should make sure that our partners are always, we are there by the side of our partners so that their careers, their life can also flourish. And when given the chance, the women in every sphere of our life have actually matched the men, and if not done better than the men. And I'm very happy for that. So we have today two ladies who have reached an area which is huge, which very few men can also reach. Dr. Sharmila Bapat and Dr. Manjari Bakre. Both of them I have known over the years, and I have immense respect for both of them. And on that talk, you will understand how much they have gone through to reach what they have reached today. Today we have got them, and we are very fortunate to have them. And I thank Kartiki, whoever, where is here, there, for helping me with the choice. So, friends, again, I will sum up. I will not stand between you and our speakers today. Just make a pledge within yourself that you will help your partner in every aspect of their life. And for the girls who are sitting there, do not give up your dreams. That is also a pledge which you have to make to yourself. Do not give up your dreams, even though there are pressures which may come from the society and from the males. Without that, without you being strong, nothing can happen. Thank you.
May I now request Dr. Mahua Malik to kindly introduce our first speaker.
Thank you, Arunima. Hello, everyone. So it's my privilege to introduce our today's guest and speaker, Dr. Sharmila Bapat. Dr. Bapat is currently professor at the National Center for Cell Science in Pune. There, she leads a research group in ovarian cancer biology. Her research interests span cancer stem cells, epigenetic changes, and network modeling in ovarian cancer. Her team has recently developed a method to profile the cells in tumors, which enhances the effectiveness of cancer treatment. Dr. Bapat has received several awards throughout her career, and she is also an elected fellow of the Indian Academy of Science, Bangalore, and National Academy of Science, Allahabad. So she has published several book chapters and several research articles in different peer-reviewed international as well as national journals. She has also served on the editorial board of several journals and as the editor of books, and also works for many research task force and academic bodies in India. So without further ado, please welcome Dr. Bapat, and I request her to deliver today's talk. Thank you. Pointer is not working. Sure. Thank you.
So thank you, Dr. Mahua, for that very kind introduction. And thank you, Professors Sengupta and Desai, for the kind invitation to be here. And again, Dr. Sagar Sengupta, for the very encouraging words. It's really rare to have a director who speaks from the podium and from his heart as much as he would. I really welcome this invitation because it was an opportunity for me to actually look back and look at and think about the people who have supported me throughout my transition into an independent researcher. And it's with a great sense of gratitude that I look at them and I think about them, because the one thing that stands out amongst all of them is that they never question me and my capabilities as a woman. I think that sense of equality that gave me the strength to think as much as any of my male colleagues would. In fact, there are several of them with whom I've shared a love-hate relationship simply because of the strong criticism they brought into my research, but never into the fact that I was a woman who was trying to do crazy things, what they thought at that time, some of which have failed, of course, but equally number of things that probably worked out. And I think you need that type of a neutral, unconditional support, which is going to question your moves, but not to the extent of deflating them. So these are the people I call as my knowledge enablers. And I think this talk is going to be dedicated to them. And I'll leave it to you to judge whether that unconditional support was worth it or not. So I work at the National Center for Cell Science, and I began my lab in 2001. At that time, there were no research labs in India working on ovarian cancer. So when I was presented with an opportunity to become an independent investigator, I thought of two cancers. One was ovarian cancer and one was pancreatic cancer, because these are the two ones that really were aggressive, and they are still recognized as being challenging, not only for researchers, but even at the clinical level. So I chose to go ahead with high-grade serous ovarian cancer, which is a subtype of ovarian cancer that is known to be very strongly associated with a high degree of mortality. Even after all these years, there are very poor diagnostic tests for their early detection. And although initially a lot of patients go into remission, there's a lot of recurrent disease, and which is highly aggressive in nature. So over the years, we've actually been able to identify a few features that are associated with ovarian cancers. The initial studies went on to identify cancer stem cells and epithelial to mesenchymal transitions, which almost went hand in hand. And in fact, in most of these, most of the work in my lab is actually serendipitous. We just stumble onto something and we just pick up these leads and try to make something out of that. And I think that is the feature that is so important for a person who's a researcher, because it gives you an opportunity to be creative, at the same time try to make some sense and apply it to societal needs. So all of our work has been almost serendipitous, our identification of cancer stem cells, EMT, then of course it was with a very directed application of the omics technologies to understand what we were actually getting in our experiments. And then try to make sense of the omics, we stumbled onto tumor heterogeneity. And currently we look at intra-tumor heterogeneity, wherein we've resolved a tumor cytotype. Then we look at phenotypic plasticity. And of course, our identification of the molecular subtypes was something that really was very satisfying, because it was validated in the clinic. And recently, over the last 5-6 years, we've been working with something called as chimeric transcripts, which are transcripts that originate from more than a gene, a single gene. And that's really exciting. And I hope I get time to talk to I walk you through all these things. So in July, in 2001, when I started working with these primary tumors, and I had a lot of clinicians who actually supported this study. The reason I started doing establishing primary cultures and cell lines is because the NCCS repository had only one cell line at that point of time from ovarian cancer, and I was establishing a lot of lymphoblastoid cell lines, and then I diverged into establishing some more ovarian cancer cell lines for my own work. So it was one of these primary tissues, OT5, it was my fifth tumor that I was processing, when I realized that there were a really, it was actually dominated by a bunch of very shiny, very aggressive looking, very healthy looking cells. Now, when I went back to the clinical characteristics of the patient, I realized that the patient had actually passed away within 15 days of us getting the sample. And I said, okay, these shiny cells must be the really aggressive ones in the tumor because they were really very dominant in the tumor. But to my utter despair, what happened was within a month, these so-called shiny cells were overgrown by the tumor stroma and we just lost them. Now my PhD had been in plant biotechnology and I had been looking at something called as somatic embryogenesis, which is a process by which the somatic cells in a plant are reprogrammed and they can give rise to either organs, but somatic embryogenesis is when an entire bipolar embryo develops from a single somatic cell. So the concept of totipotency was always there at the back of my mind and in my training. And I actually failed to understand why animal cells could not just grow. Forget them being totipotent, but at least they should be able to grow, especially if they came from tumors. So then I thought that, okay, that was also around the same time when Professor Dick in the University of Toronto, the Sick Kids Hospital in Toronto, had identified leukemic stem cells based on the common markers that are expressed by primitive normal hematopoietic stem cells and their expression also in tumors. And he went on to show that these small rare groups of cells which share the same primitive stem cell marker could actually give rise to an entire disease in immune compromised mice. So it would have been easy if the normal ovarian cancer stem cells or the markers of ovarian stem cells had been known, but again there was no report of that. And also I was also motivated by a review that had just come up in the very year, which is now a classic review by Rhea Tanishta, and which postulated that there are likely to be stem cells even in solid tumors. So driven by this, I postulated that probably at around the same time, we were also bringing in normal ovarian samples, human ovarian samples. And what we found was a process called as epithelial to mesenchymal transition, which was associated with the surface epithelium, which was thought to be the cell of origin of ovarian cancer at that time. And we went on to study this also on the side, and we actually associated some of the transcription factors that drive EMT to drive metastasis in ovarian tumors. This is something that we did in the primary tissues. We found it even in the single cell line that we had in our repository and in our primary cultures also. But coming back to the cancer stem cell study, what we thought was that could we use the long limiting dilution approach in the absence of markers to try and isolate these stem cells. So what began was almost 2 years of very laborious single cell cloning from primary tumors. We established a large number of cultures, and we actually first were quite lucky with one sample wherein we had about 70 or so single cell clones, but unluckily we lost them all during consecutive passages. Finally, was successful with this approach in 2003 April with a sample that came from a very advanced stage ovarian cancer patient. And through this, we actually did single cell cloning, and we established 19, we had hundreds of single cell clones that we had established, but out of these 19 actually underwent a spontaneous immortalization event. That was also the same year that the first publication in solid tumors by Alhaj was on breast tumors, and following that Sheila Singh's group worked on neural tumors. So that enthused me because I knew that we were still working in the right direction. And our work, we went on to work out on these 19 immortalized clones and went on to show that these have a lot of stem cell characteristics and that they could contribute to the aggressiveness of ovarian tumors. Very briefly, this is what the study was about. Now, in high-grade serous ovarian cancer, the patient has peritoneal ascites that is rich with three-dimensional tumor spheroids, as they are called. So from several spheroids, we actually cloned single cells we had 65 single cell clones that seemed to be capable of being immortalized, but out of these, only 19 of them could be continued beyond around 15 to 20 passages. We then extensively characterized these clones for the expression of pluripotency markers, cell-specific markers, as well as differentiation markers, especially in the spheroid cultures, and then further characterize these parameters in animal models. So these were the set of principles that we set down in 2005, wherein we could correlate some of these functional assays with a stem cell-like character, because that is something that was not really very clear in the field at that time. And then the limiting dilution assays, wherein we actually inject a very low number of cells in immune-compromised mice, and we find that only some of these can actually give rise to tumors. Thereby, the disease could be established from a very few number of cells. And the final proof was obtained by the similarity at a histopathological level in the tumors that we had generated in our mouse models and the human tumor. And also the pattern of metastasis that was absolutely identical to what is seen in the human disease. So this conclusively, for us at least, established that these clones are likely to be stem and progenitor clones that would very faithfully perpetuate the disease in animal models. Now, once we had these 19 clones, what I forgot to mention earlier was that there was only one of these 19 clones that was actually tumorigenic and could generate tumors in immune-compromised mice. Another clone actually acquired this feature of transformation during culture and became a tumorigenic clone. But that meant we had 17 non-tumorigenic clones and just 2 tumorigenic clones. So we were wondering what those clones represented. Now, around that time, Bob Weinberg was actually trying to create a artificial trajectory towards transformation by knocking in, say, one oncogene at a time or one mutation in RAS at a time, as well as knocking out some of the tumor suppressors and trying to find out the threshold at which a normal cell could be tipped into transformation. So, I mean, we thought that, okay, probably we have just picked up something unknowingly, which is a trajectory, and which might have all these different mutations poised in the trajectory towards transformation. Now, I'm talking of around 2004 and 2005 when there was no RNA sequencing. And so we had no tools actually to study the genetic or the mutational landscape. So what we thought was that let's look at the mitochondrial mutational landscape. Now, the mitochondrial DNA, as you know, is a very small DNA, and it can actually just be amplified in totality. So we did that, and to our surprise, what we found was that 5 out of those 19 clones could be predicted to have the same mutant, or rather they grouped together, although 3 of them were non-tumorigenic. They grouped with the tumorigenic clones, whereas 14 of them maintained the germline profiles of mitochondrial DNA as the patient. Now, this was surprising because we had thought that all of them are literally transformed cells. And having a germline profile meant that they were really not anywhere in the associated with even the transformed clone, but they were probably supportive cells associated in the spheroids with the cancer stem cell clones. So we started profiling these for markers, and at that time CD133 was largely being touted as a cancer stem cell markers, and I think there are a few hundreds of papers in which using CD133 researchers have isolated cancer stem cells and gone on to show at a functional level the regenerative potential. But in our case, CD133 was actually expressed by the non-tumorigenic clones. So cutting a long story short, what we found is that the cancer stem cell clones actually recruit the CD133 cells, primitive stem cells, towards establishing a tumor vasculature, which we showed in the in the matricial tube formation assay. as well as we also showed the entire endothelial lineage or the hierarchy that develops from these CD133 cells. Sorry about that, excuse me. One very nice study that Anjali Kusumbe, who was in my lab, who was a student in the lab at that time, did, is that she tagged some of these CD133 with a GFP marker, introduced these into mice through the tail vein, and showed that these CD133 expressing cells actually home to the cancer stem cells, which may be in the peritoneum and giving rise to ascites, or the primary tumor. So this is the primary tumor to which the CD133 cells have actually, the GFP positive cells have home towards, as well as the tumor ascites that harbored these spheroids. So this was a very satisfying study for us. Now, as I mentioned, I had some enablers, knowledge enablers, who were seriously criticizing my work. And one of them just posed to me the simple question, saying that, Sharmila, you know, we've been working with cancer cancer cell lines for decades. She was a very senior researcher, and she said, we've been working with cancer cell lines for decades. We show that they form tumors. We went in mice, and in culture, they're virtually immortalized. So what is the difference between your cancer stem cells and what we just call as cancer cell lines? So this was very serious criticism, but it was put in a very constructive way that made us think. And the one thing that we said that not just us, but the entire cancer stem cell field was missing was a demonstration that these cancer stem cells can stay quiescent. Now, quiescence is a feature that distinguishes stem cells from a normal somatic cell, simply because a stem cell can stay dormant and then it is activated by the microenvironment into a state of division. And when it divides, it's an asymmetric division whereby one cell that is formed goes on to become the workhorse of the degenerative hierarchy and it replenishes the tissue, whereas the other cell, it goes back often to the same niche and stays quiescent again. So what we and the others had not demonstrated in the field was this phenomenon of quiescence. Now, it's easy to, it was relatively easy, let me say, to identify this as a gap in our study, but we wondered about how to do it. And finally, we hit upon a trick. What we did is that we labeled some of these cell lines with a vital dye. It was a lipophilic dye that labels just the cell membranes. So what happens is that when the cell divides, it will dilute the dye equally between its daughter cells, and this will continue until the dye is entirely quenched out. So cells that retain the intensity of the dye are the ones that are likely to either not have divided at all or have undergone just a minimal number of divisions. And so what we did is that we labeled the cells and generated xenograft tumors from these. And then after several weeks, we harvested these tumors and then subjected them to flow cytometry based analysis. So what we saw is that these tumors had cells of varying dye intensities, a very small fraction of cells that had the same intensity as the cells at the time of injection, which suggested that they had undergone a very minimal number of divisions. Around 25% of the tumor, 20 to 25% of the tumor that had a medium or a low level of intensity, and a large fraction of the tumor, roughly 75% of the tumor that had no intensity left at all, which suggested that it actually was generated through a series of multiple divisions in these. So the dye we used was called as PKH67 or PKH26. And so the PKH high were likely to be quiescent cells, PKH low were likely to be progenitors, and the PKH negative were likely to be the differentiated part in the tumor. We validated this postulate with using the set of functional assays that I showed you in the earlier slide. And then Of course, we could also see this differential intensities in cryosections. The red is PI-based staining, and then you have several foci which retain the cancer stem cells, suggesting that there is a specific niche for cancer stem cells even in a tumor. More importantly, we also identified that chemotherapy actually leads to an enrichment of these cancer stem cells, and that when the chemotherapy is discontinued, these could actually grow out and give rise to recurrent tumors. So this was the next level by which we actually modeled the human disease in vivo models, and this gave us a lot of understanding, and very importantly indicated that you do have a quiescent fraction of cells in vivo or in tumors, which can be called as cancer stem cells because they do have this reversible quiescence wherein they can be induced to form tumors. We would sort out the PKH high population and then put them back into mice or look at their drug resistance, and we found that both these characteristics were drastically different from what we saw in the entire tumor. Now, meanwhile, our EMT story was also working out hand in hand, and it so converged that we were actually finding an association between EMT and cancer stem cells. We had started this work in 2001, and it took us almost 8 years to realize that EMT could be one of the mechanisms by which you have an enrichment of cancer stem cells. Now, this was a study. In this study, what we had actually done is that in the meanwhile, we had identified that two transcription factors, snail and slug, were actually associated with EMT in ovarian cancer, and that these were enriched even in situations when there was no necessity of a EMT. So we started doing something called as chip-on-chip. That meant chromatin immunoprecipitation for these specific transcription factors, and then we would actually hybridize it to promoter arrays, microarrays, and then look at the promoters to which these transcription factors would occupy the promoter regions. So these transcription factors identify a consensus E box, and it was very easy to identify which are the promoters that were occupied by these transcription factors. So this genome-wide profiling showed that under steady state conditions, you have a genome-wide target of these transcription factors. Now, classically, what people were doing is looking at just the targets in, say, the cadherin junction, and E-cadherin is the flagship target of all these transcription factors, but also in desmosomal junctions as well as tight junctions. But beyond this, we had actually profiled a genome-wide target identification program, and surprisingly, under steady state, these transcription, they are repressors. They repress some of the stemness associated genes. However, under conditions of stress such as radiation, we use gamma irradiation, or chemotherapy, wherein we use paclitaxel or cisplatin, we found that they would actually lose these targets. They would acquire new targets, which would enhance the EMT process, but at the same time, they would actually upregulate these stemness genes, which led to the acquisition of a stem-like phenotype. Now, this is probably one of the, most satisfying studies that we did from the lab, because it gave us a very good understanding about why, following chemotherapy, you have an enrichment of cancer stem cells, and thereby one can start thinking about how to target these. So by this time, we had realized that whatever experimentation we do in our small studies that in our lab, they really are very small, specific, focused studies, but they are literally still working. We are still working in silos or in isolation with these studies, and each one of these studies will give us big clues as to what might be happening in the disease, but it will never be able to project onto what the entire tumor behaves like. So for that, you really needed the omics data, and that was our impetus to move on to the omics and then try to integrate some of our findings from our siloed studies and project them onto the omics outcomes and try to make sense of these. So we did a lot of things. I mean, we have done a lot of epigenetic biomarker identification. We tested several epigenetic drugs, tried to see how using these markers we could predict the outcome of chemotherapy and so on and so forth. But when it came to validation in tumors, so we had beautiful signatures. We were very happy. Outcome was that we could never validate signature molecules, and it was very depressing because often there were tumors that did not even show any expression, whereas on the other hand we had tumors that showed a very high expression. So what do you make of all this in the same disease? Is it really a marker? Can you call it as a signature molecule? It obviously can't be. But what actually made it very important to look at is look at tumor heterogeneity. We often study cell lines which are homogeneous cells, but even within cell lines you will not have an expression, the same marker being expressed in all the cells of that population. So you have to consider that there are several intangible mechanisms that would regulate gene expression patterns. Now, it would be possibly impossible to actually harness or get a grip on all these regulatory mechanisms, but we have to take into account this diversity in all the cellular, molecular and functional features that are associated with tumors. And most importantly, what we realized was that if just the validation of our biomarkers was such a challenge, then I think the real take-home message was that targeting these tumors would actually be, or its therapy of these tumors is likely to be even more challenge. Now, this to make this point more clear is that we actually looked at the response of a cell line or a xenograft actually from a cell line we had established, and we saw that when treated with paclitaxel, cisplatin or gemcitabine, you had a drastic reduction at a tumor volume and at a statistical level, which is usually considered as a very good endpoint for a drug to be considered as very effective. However, we know that the patient passed away within a month, so this was definitely a wrong readout for us. On the other hand, we had a monoclonal antibody that we had developed in the meanwhile, and which actually did not seem to be very effective. Although it was statistically significant, it still left a large amount of volume of tumor behind, yet the tumor did not really grow much. And it was the lifespan of that mouse was much longer than those treated with some of the conventional drugs. So that meant that we really need to understand what are the residual cells following chemotherapy. And we looked at it at 3 levels. One was the stem cell hierarchy identified in terms of the pKh, dye intensity, then aneuploidy that actually comes up in most ovarian cancer patients as well as in our xenografts, and then the differential cycling within the cell. So using these 3 parameters, we could resolve a xenograft into 18 different cellular subpopulations demarcated based on the cancer stem cells, their progenitors, which may either be euploid or aneuploid at the different phases, and then the large differentiated part of the tumor, and the host cells that are actually are usually a component of the TME also. Now, very interestingly, when we looked at the targets of each of these individual drugs, what we found that each drug can effectively eliminate only certain populations. Now, this provided an opportunity whereby we could define the combination of drugs that would effectively be good enough to eliminate the entire tumor. We tested several combinations, and what worked the best was the monoclonal antibody with 5-AZA-DC, which is a demethylating agent, and gemcitabine, which is actually in the clinic, and it targets proliferating cells. We took a batch of mice through 6 cycles of chemotherapy that is usually received by a patient, and these 3 drugs in series. And what we had at the end of the treatment was a tumor that was totally retracted. And lifespan was increased from 52 days to 150 days, wherein because of the animal ethics requirement, we had to actually terminate those mice. Now, one of the reasons why this chemotherapy or this combination worked was that the target of the demethylating agent 5-AZA-DC was the target molecule that is recognized by the monoclonal antibody. So thereby, this annexin A2 was the target, and it has a CTG island in its promoter. Thereby, by treatment with 5-AZA-DC, we were actually upregulating the targets of the other two combinations. So this, in part, was very useful for us to know, because it gave us an understanding about why the treatment was also effective, and that it was not likely to be just a chance event. So this was again very important to know. Now, this was fine about the intra-tumor heterogeneity that is reflected as the different cellular populations within a tumor. Then we started thinking about the different tumors from different individuals or different patients. And we realized that at that time, people were talking of inter-tumor heterogeneity. And of course, breast cancer heterogeneity is very well documented and studied with targeted treatment in place. So we wondered whether we could try and develop such a similar approach, and we did that. The TCGA had actually selected ovarian cancer as the second tumor of choice for the entire profiling, and this actually gave us a huge opportunity for us to integrate into our research. So using the RNA sequencing data from TCGA, we did something called as a weighted genes weighted correlation gene network analysis, and we came up with these modules of correlated genes. Now, what we found is that using these modules, we could actually segregate 3 different groups of tumors of patient samples, and we called them class 1, 2, 3. And as you can see, class 1 and class 2 seem to be totally anti-correlated. So whatever was upregulated, which is in red, or these are each square of these tiny square is a gene. So all these genes that are upregulated in class 1 are classically downregulated in class 2, and vice versa. We went on to do a lot of network analysis. We looked at, we identified cell lines that are representative of each of these classes, went on to show the biological functioning that gave each class of tumors its own functional identity. And what we found was that one of the classes was dependent on EMT-based invasion, whereas the other class, surprisingly, could not undergo EMT. The cell models that we were using, they just would not undergo EMT. When we even tried to express some of these EMT transcription factors in these cells, they would undergo apoptosis, and they would never survive. Now, Sagar in the lab Sagar was a student in the lab at that time, and he went on to show that these models of class 1, the cell models of class 1, undergo something that is called as cooperative cell migration. That is, they maintain an epithelial phenotype, and they actually migrate as clumps of cells or clusters of cells. And he has done beautiful time-lapse videos wherein you could see that EMT, or the cell lines that are capable of EMT, actually finish off wound healing within anything ranging from 16 hours to 40 hours, whereas the class 1 models would not achieve wound healing even after 120 hours of live capture. And the only way they could fill into the wound is through a rapid proliferation rate, whereby they would just mechanically slough off excess cells into and try to fill up the wound by colonizing in the area that was vacant. So this was very nice to identify, but we wondered. Now, around that time, there was also a lot of criticism coming worldwide that EMT is actually an artifact of in vitro culture. And this was basically from pathologists. And in fact, there was an entire session in AACR annual meet that year that was presented by pathologists all over the world who very strongly refuted the occurrence of EMT in situ, that is in a patient. So what we said is that, okay, if we have a class of tumors that is suggested to be enriched with a mesenchymal phenotype that arises through EMT, we have to find out if it exists in patient samples also through pathology. So we actually got together a team of pathologists, And we went on to identify 6 markers of the, which were specific for these 2 classes. And we showed that even patient samples, Swapnil was a student in the lab who actually worked with 96 cases of high-grade serous ovarian cancer. And it was a total of 146 samples, I think, that he really looked at. And we had a blinded review by all these pathologists. And what we came up with is that, yes, even in situ, you do have class 2 that is identified. And further, we actually, the immunohistochemistry also helped us resolve the slightly confusing class 3, which was a mixed type of a class, into double positive, that is those that express both the features of the 2 classes, or double negative, wherein probably these markers were not sufficient to stratify those. So this meant that definitely that these classes do exist in human patient samples also, and that was when we decided to take up an active targeting program. We looked at annexin A2 and we developed a monoclonal antibody against that, and we've shown that it is effective in treating the xenograft models that are representative of the class 2 samples, which was the data I showed you earlier, and that worked very well in combination with 5-AZA-DC and gemcitabine. So this actually gave us an opportunity to not only just stratify tumors into their distinct phenotypes, but also design therapies that would target a certain group of tumors. So MAD 150 is probably going to be of no use in targeting class 1 or class 3 tumors, but it would be effective only in class 2 tumors. So this is the monoclonal antibody, and as you can see, this was a PDX model that we had also developed, and which had a high expression of annexin A2 in its surface, and we could actually eliminate the entire tumor in a PDX model. That was very satisfying again. Now, do I have time left? Okay, so this is something that is just going on in the lab at present, the last 5 years. The impetus for this was that everywhere immunotherapy was being thought of as probably the next level of therapeutics for cancer, and which hold a lot of promise. At the same time, there was a lot of literature coming up saying that ovarian cancer is likely to be is actually not considered a very good candidate for immunotherapy, simply because they have a low mutation burden. 95% of ovarian tumors or high-grade serous tumors have P53 mutations, which are largely undruggable. Around 20% of them have BRCA1, BRCA2 mutations, for which there is already therapy in the at an advanced preclinical stage. Wherein you have PARP inhibitors that have been shown to be very effective against the majority of the patients. But apart from this, you have hardly any mutational landscapes in ovarian cancer. Yet, when we were doing our immunohistochemistry, we saw that there were certain tumors, or certain patients had tumors that were enriched with a large number of infiltrating T cells, and these patients invariably had a better prognosis also. There were also there was just 1 or 2 additional reports apart from our observations. So we said that probably there are going to be some non-canonical neoantigens that are generated. And one crazy idea we had is that why don't we look for translocations? And translocations are nothing but new transcripts that are generated from 2 genes. So we decided to use the RNA sequencing data on a cloud-based platform, wherein we looked at chimeric genes, and we identified almost 4,000 of these in 161 tumors, which were impossible to be translocations. So this meant that there are quite likely RNA transcripts that are generated that seem to be chimeric in nature. We've been doing a lot of, what do I mean by RNA chimera, is that, say this is a 3' gene and this is a 5' gene. This is the direction of transcription of this gene. And what happens is that you have some of the exons of the 5' gene, and very often a read through to pick up an exon from a downstream gene. And this read through mechanisms is most largely touted, although we also identified different other mechanisms, including chromatin compaction, that may be responsible for the generation of these chimeras. So this actually started off a big program in our lab, wherein we are looking at novel splice variants. We're looking at some of these sequences of these chimeric transcripts actually match some of the pseudogenes that are already there in databases. So we have another project that we are looking at in this. But what was most interesting, and again it came up as a response to criticism, was that I had criticism that these are nothing but artifacts of RNA sequencing. So I said, okay, but I'm not the first person to report this. This has been in the literature. That was my defensive response. But then what I thought was that going ahead, we should probably try to detect if any of them are actually translated. And to do that, what we did was that we made a customized database wherein we performed an in silico translation of these, the sequences of the transcripts we had, and we provided that as a backend database for mass spectra data. And through this, we started picking up our chimeric transcripts. For example, this is just one example wherein you can see that these are the residues that originate from the 5' transcript, and these are the residues that originate from the 3' transcript. So this was a fairly good proof of principle that it was, I mean, we were bypassing the artifacts of RNA sequencing by just generating peptide or identifying peptides. So we could very well address that. We then looked at some of these peptides and tried to predict whether they have any neoantigenicity. And this was just a computational pipeline that we use. And this is quite a fairly standard computational pipeline. And in totality, the take-home message was that we could identify some specific antigens which were not present in normal tissues, but were generated through this chimerism and which are likely to be recognized by the PMC molecule, the MHC1 class of molecules, and then presented to TCRs. And this was a molecular docking study done by Vaishnavi in the lab, and she effectively had to screen. It was like finding a needle in a haystack, wherein she looked at what were the PDB structures available for MHC molecules, TCR receptors, and of course the prediction of our peptide. And then she found a few neoantigens that are likely to elicit immunogenic response. So Ritika in the lab is now taking this study ahead with a syngenic mouse model, and we hope to be able to take these further. So with this, I hope I've been able to convince you that my knowledge enablers were justified in supporting all my crazy ideas, and we've been able to do a lot, but there's so much more that still remains to be done. I thank you all for a patient hearing, but I have to really thank all the alumni who've been working towards their degree or towards just a dissertation. And I have a really bunch of talented students currently in the lab, and every time we have a new entrant, a caricature comes up on a whiteboard. So this is me, believe it or not. And then we have a new person who goes on the board within a week of their joining the lab. So it's really very enjoyable to see this happening. And what remains as a huge motivation for me is a person, Laurel Pratt, whom I met in 2014. At that time, she was a 15-year survivor of high-grade serous ovarian cancer, which is a very long-term survivor. I mean, this is the longest lived survivor I have met. And even today, she communicates with me. She keeps on asking me. She keeps me on my toe. Sharmila, is there something new you're going to give us? So that really keeps us on our toes and provides an impetus for further thought. Thank you all, and I'll be happy to take questions if there are any. Sure. Thank you.
Thank you, ma'am, for your talk. I would like to call you back on stage, and Director Sir, can you please come on stage to felicitate ma'am for her talk? Ma'am. the left. Thank you, ma'am. Thank you, sir. Now I would like to request Professor Kartiki Desai to come up on stage and introduce our next speaker for the day.
Thank you, Dr. Bapat, for that scintillating talk, and happy Women's Day to everybody in the audience. We are a day late because of holy celebrations, but our enthusiasm is the same. So let's move on further for today's talk. And it is really a very exciting thing for me to introduce my friend and Manjari Bakre, who is only 6 days older than me. 6 days older than me, so we share that. It's the same fire of that sun sign. And nevertheless, I would do a slightly informal introduction because she's been my friend for many, many years. We studied together in Indian Institute of Science, and we belong to the same department. And we have just followed each other's careers for a very long time. So she did her PhD in Professor Sandhya Vishweshwaraya's lab, and we had a lot of interaction at that time in which scientifically also interacted with each other. Then she moved to Mount Sinai to do her postdoc, and then she moved to UCSD, and she has a fabulous Nature Medicine paper, and then probably that's where from guanylate cyclases and cholera toxin she got into the field, which is again overlapping for both the speakers, which is embryonic stem cells as well as cancer stem cells. Then she moved to GIS, and I almost followed her there eventually, because when I got an opportunity in Singapore, I was like, Singapore is an island, why would I go there to work? But then Manjari was there, so she helped me make the decision. Although I joined another thing, she was already there in GIS. So she again worked on embryonic stem cells over there, and she has a JBC paper which is got the best paper award. So that is only the top 1% of the papers actually are given this honor of being the best paper award with JBC. So I just wanted to describe her scientific career, and despite such a stellar scientific career, she's the brave one who jumped into industry. And since then, I have learned so much about industry from just Manjari actually. So if I ever want to, she also helped me decide probably it's not for me, I am a better academician, was one decision. She's helped me, I guess, to make the lab, but I'm also very happy that she's been very, very successful. She's worked very hard. She had a very small stint when she came back to India in Philips and Avastha Jain, probably to learn the roots. And then she decided, that's it, I'm going to go independent. She founded her company, if I'm not mistaken, it was 2011, called OncoStem Diagnostics. And now she is very, very successful in having a breast cancer test, which she is marketing. And she's going to talk to us about her journey, about how she established, she's founder and CEO of OncoStem Diagnostics. And I'm very, very happy to have her.
Both of the videos are already inside your PowerPoint also. I have put them inside your
Okay.
PowerPoint.
Yeah,
Sure. This.
Okay.
You have given two empty pages inside
Correct.
that. I have put
Okay.
them there.
Sure. Thank you. Thank you all for coming here, and thanks Kartiki for the introduction. Really appreciate invitation from you, Dr. Sagar. It's always a pleasure speaking to friends and colleagues. I met Dr. Sharmila in a conference in Jaipur. I remember that was this thing, and so I mean, as I know, I was just wondering, when I heard Dr. Sharmila's talk, like, I'm not going to show so much science. so it's glad that you guys have had enough of science from her. So my talk will be very different. So I actually didn't know what to speak, but I thought since it's Women's Day, I thought I would also put in some flavor, some color on what it takes to really, follow your dreams as a woman, and for those who have ambitions to do different things, whether it's entrepreneurship or it's something else, feel free.
And Kartiki, thanks for the introduction. It was not as bad as I thought, so I'm happy.
I didn't have the secret.
Correct.
Now everybody wants me to give out mine.
Yes, yes. We'll have a long session on that. Okay, so all right. So coming to this, the talk for today. So I really wanted to explain from scientist, my journey as a scientist, from scientist to an entrepreneur, and how did OncoStem evolve. So the idea of OncoStem really, I'll give you background behind that, and then I'll tell you what product we do, we have, and what else we are working on, and then the journey of the company. So when I was doing my PhD, I had a senior who had breast cancer, who felt the tumor herself and was diagnosed early stage. But she went ahead to do her postdoctorate fellowship to Israel and passed away there. So that made me think, when tumors are small, how can we not know how would the disease progress? If the tumors are big, patient has few years, months to live. But breast cancers, especially when tumors are small, one assumes that they have good survival, but she passed away. And if she knew that she was the only daughter, and she could not even be brought back. So it really made me think, why can't we prognose a patient's disease, especially when tumors are small? And that seed was there in my head. So that really led to OncoStem journey in the end. So just first, only one slide on the company really. So OncoStem's mission is to really develop innovative prognostic tests to personalize women's cancer treatment and improve patients' quality of life with reduced cost of care. So, in other words, we are not into screening space. Prognostic tests are tests which are performed when a patient has known cancer. And we are only helping doctor and patient understand how aggressive is the cancer and how to treat the patient post the diagnosis and confirmation. We are not into screening or diagnostic space. And our first product, which is CanAssist Breast, it's a prognostic test for breast cancers with hormone positive, HER2 negative disease, and it's one of the first made in India prognostic tests. So, what image does cancer often flash in our minds is chemotherapy. Cancer is almost synonymous with chemotherapy, and very few patients who with cancer really are spared of chemotherapy. I want all of you to focus your attention only on breast cancer. But does every breast cancer patient really need chemotherapy? And there have been a lot of studies on chemotherapy. How it started was during the World War, how it was really discovered the power of chemotherapy during the World War with nitrogen mustards, and then really the pendulum, everybody was over-treating patients with chemotherapy to cure them, and then they realized they were over-treating, and then started the randomized prospective trials to really understand, is chemotherapy required, which chemotherapies, how much, etc. So all of that really came up later. So standard treatment for cancers have been, and until this World War II, until the 70s, 60s, it was really the domain of surgeons. And then local treatment really means surgery that is local to the site. In case of breast cancer, it would be surgery and radiation, and systemic treatments would be hormone therapy, which is a tablet you take per day, chemotherapy, which is IV, and targeted therapies like Herceptin, any anti-HER2 therapy would be targeted therapy in breast cancer. So every patient need not go through all of the standard processes is what is now a realization, and maximum everything would pump the patient with every drug is not what's going to help the patient, but minimal treatment with maximum benefit to patient's overall health, not just the cancer, but the whole patient. You have to look at the patient in as a whole is really what is important. So there is really no one size that fits all. There is no one treatment that you give, one kind of procedure that is local and systemic therapy that you need to give it to every patient. So in case of breast cancer, there have been multiple studies which have shown that actually only 15 out of the 100 patients when you treat with chemotherapy actually benefit from it. And this, I'm only talking about hormone positive, HER2 negative, early stage breast cancer patients. Early here refers to T1, T2 tumors, that is up to 5 centimeter tumors with up to 3 lymph node positive tumors. So these tumors are small, and in breast cancer, you can feel the lumps, you can get them diagnosed early as against many internal organ cancers. So when that is the situation, when a surgeon can resect it completely with leaving clean margins in the body, there's really no need to over-treat the lady with all the kinds of chemotherapies that we typically give today. But the clinicians are faced with this dilemma. They don't know whom to treat and whom not to. I mean, just because if there are 2 patients with 1 centimeter tumors, it really is never a case that both will recur, and it's never the case that both will not have a recurrence. So how do you know which of these 1 centimeter patient tumor will have a recurrence, which versus not? And that is what is prognostic tests help you to find and really optimize the treatment, especially chemotherapy. So prognostic tests in short help to find these patients who have a high risk of breast cancer recurrence, and only those patients should take chemotherapy, sparing the remaining who have a low risk of breast cancer recurrence. The font has changed, but never mind. So prognostic tests are, as I mentioned, for only, I'm talking with respect to hormone positive, HER2 negative EBC, early breast cancers. They are done on post-surgical samples, whether it's lumpectomy, mastectomy, breast conserving surgery, anything. And they typically segregate patients into 2 buckets, those who have a low risk of breast cancer recurrence, and that risk of recurrence we measure from the time of diagnosis up to 5 to 10 years from diagnosis. And those who have a high risk of breast cancer recurrence, who should take chemotherapy. They should not only take chemotherapy, but any other targeted drugs. This is HER2 disease, so obviously there is no other targeted therapy apart from hormone therapy. But CDK4/6 inhibitors, maybe if they're BRCA mutant, then maybe all the PARP inhibitors and so on and so forth are IO therapies that are out now. So in the West, prognostic tests have been, so the only competition we have, Oncostem has, is from the West, Oncotype DX and MammaPrint from the Europe, Oncotype from the US. And these have been really standard of care in those countries. They are in every important guideline, NCCN, ESMO, NICE, ASCO, AJCC, etc. And they have definitely led to reduction in use of chemotherapy for early stage breast cancer patients in Western countries, improving the quality of life of the patient. So, but recently they have TAILORx and RxConDo were the biggest trials, prospective randomized trials that US conducted for Oncotype DX, and they have realized that not all tests work across different ethnicities, because the way they were developed, the way the kind of biomarkers they look at, etc. So especially MammaPrint, Oncotype DX was shown to work differently in African Americans versus white Caucasians, and Hispanics versus white Caucasians. So they do have a slightly aggressive phenotype, probably the African Americans or Hispanics, and therefore the tests work differently. So what is essential is they have concluded is to really validate, either develop the test on that population or validate it on that population and titrate out the scores or whichever methodologies that are helpful. So that is where I saw an opportunity, and so why I wanted to develop Can-Assist Breast is because of this friend who died of breast cancer recurrence, and Oncotype is beyond our patient's reach. It costs $4,000, no matter whether it is performed in the US or in India or in Vietnam. So nobody in India, not every patient can afford to do $4,000 test, and then if you are high risk, then you also pay for chemotherapy. So when I worked with clinicians throughout previous engagements in India, and I used to ask them how if I develop a test like this, will you use it? And they say yes, we will, because I know I'm over treating some of my patients, but I don't know whom to spare. So by default, I give chemotherapy to everybody. And I said, so why don't you use Oncotype? And they say it's very expensive. So that really made me feel that we need a test which would really work for our patients in all aspects, not only the biology wise but also cost wise. So Can-Assist Breast is a test that helps to find, it's a prognostic test which helps to find the 15% patients with aggressive tumors, and we use immunohistochemistry as a technique and AI/ML based methods to segregate patients. So once again, Can-Assist Breast is performed on patient's tumor that is resected during surgery, and then 70% are considered to have a low risk of breast cancer recurrence based on a risk score that we get from our algorithm. I will get to those details, and 30% do have a higher risk of breast cancer recurrence, and therefore will benefit from not only taking chemotherapy but better follow-ups, better additional targeted therapies, etc., that they can get. And Yeah, as I mentioned, intended use is only for hormone positive, HER2 negative, early stage breast cancer patients. And we have been selling Can-Assist Breast for last 6 years in India since middle of 2016. That's when we started selling. And this is a patient who has actually used Can-Assist Breast in very first year of our sales and has benefited. And this is a real patient we follow every year. So this Alpana, name change of course, was 31 years old in 2016 when she was recently married and wanted to have a baby but was diagnosed with breast cancer. So what should she do? So her wish was to have a baby, but at this age, 31, and she had T2 disease, 2.5 centimeters, lymph node negative but high Ki67, grade 2 disease. So with her age against her, I mean, typically we consider these as kind of empirical factors, clinical parameters, which are important in predicting risk of breast cancer recurrence. So she would have got entire suite of therapy, that is surgery, radiation, chemotherapy, and hormone therapy. And hormone therapy until recently was given for 5 years. Now it's 7.5 years. So, and she can only have baby after finishing hormone therapy, which will be minimally 37. And then of course she needs to wane, she needs to be weaned off of the hormone therapy. So she's looking at least 38, 39 to have a baby. So she has to either go for fertility preservation, etc., and then have baby at 37, or the other option, only other option that her doctor suggested is do this test. Let's assess your risk of breast cancer recurrence and then plan therapy. So when we got her tumor tissue sample and we ran Can-Assist Breast, we predicted her risk of recurrence to be low. And so that's the report we gave to the doctor, and he actually went ahead and skipped chemotherapy entirely for her. He allowed her to have baby first, right after surgery and radiation. And post-delivery, she is put on endocrine therapy, and she is a happy mom of a 5-year-old baby now. So this was really one of our first patient who was doing well for the last 5 years. So this just goes to show that you need to really think out of the box. You need to, what needs to come first is the patient and patient's wishes. And I'm really happy. It's every time I talk about this patient, it's really heartwarming because I remember seeing her, and it's where she is now. She is definitely in a happy place. So having said that, what is Can-Assist Breast? This is a test which is performed on tumor tissues received from the patients. These are received as formalin-fixed paraffin embedded blocks. We do immunohistochemistry for 5 biomarkers, and those are graded by oncopathologists who are trained in our lab, and this is only performed in our single central lab, which is CAP and NABL accredited in Bangalore. And that input from oncopathologists along with patient-specific tumor parameters, which are tumor size, tumor grade, and the lymph node status, that goes into our machine learning-based algorithm, which is again developed by us. And we get a risk score, which we call Can-Assist Breast risk score. It's a number between 0 and 100. If the number is 15.5 or lower, we consider the patient to have a low risk of breast cancer recurrence, and she can potentially avoid chemotherapy. And if this risk score is higher than 15.6 up to 100, we consider the patient to have a high risk of breast cancer recurrence, and typically the segregation is about 70-30. And so far, Can-Assist Breast has been validated in different geographies. In India, it's primarily developed. 95% patients are Indian origin. We do have 5% Caucasians. And test is also validated in US in 3 sites and in Europe in 5 different countries. And it does have a high accuracy of 95% negative predictive value, which is how we measure accuracy in prognostic tests, which is comparable to any Western test. And the patent for this 5 biomarker combination is granted in multiple geographies. And in today's day and age, you can't survive without scientific peer-reviewed articles. So this is where we have done all the validation studies in 3 sites in US, 5 countries. Netherlands is missing here, and so many hospitals in India. So why Can-Assist Breast? It has multi-country validation, which is, the major difference is Oncotype DX between us and this. It is developed and validated on younger patients, and our patients get the age at diagnosis is premenopausal. Median age is about 48, and in West it is postmenopausal beyond 50, 52, 55. It is an affordable innovation. It is 4 to 5 times lower than the competitors. Cutting edge, it uses cutting edge AI ML based competitors, and we really spend time and effort in really developing this algorithm. to really make it robust, to beat any ethnicity, to really look at the tumor biology and come up with the biomarkers. And then it is quality driven. You can't do anything in healthcare without any quality, CAP, NABL, ISO. We also have CE IVD mark for this thing. We are working on transition to IVDR. And the turnaround time. So our turnaround time is just 7 to 8 days, whereas if you have to do oncotype, the sample has to go to the US, so it takes much longer, about a month's time. And since 2016, we have been selling Canasys breast across India, Sri Lanka, Bangladesh, and of late last 2 years in Turkey and UAE, Middle East as well. And more than 3,000 patients have benefited by running Canasys breast on their tumor tissues, and based on that, plan the treatment. Typically 75% of these patients have been classified as low risk for breast cancer recurrence. And interestingly, we have been really working hard to do this task. It's a task that takes half my energy and 10 times the time I have to get insurance reimbursement from Ayushman Bharat and any other health and state government schemes, and also from private insurances as well as public. So, but it's heartening to see that many patients who have insurance coverage, private or public, when they apply, they have been reimbursed with the price of the test. So that is very heartening. But we want it on for every patient, and this is currently done really ad hoc, case by case basis. Some patients get and some don't. So we want to really get into Ayushman Bharat scheme of breast cancer treatment. So it would be done routinely for every eligible patient and would be reimbursed to them as well. And here is once again a patient who is given us testimonial, another patient who says, today I am symptom free over 5 years. She was also low risk for breast cancer recurrence and has gone back into her kitchen and everything and is really enjoying. And with respect to cost also, so many, like this is an auto driver's wife who took the test and has given us this testimonial. This patient had breast cancer 10 years ago and she took chemotherapy because there was no such test. And then now she had, in 2017, she had breast cancer again. And that's when now she has taken Canasys breast and did not take chemotherapy. So she is also benefited by avoiding chemotherapy the second time she had breast cancer. So now comes the scientist to entrepreneur part of the talk. So as Karthikeya mentioned, in 2011, when I was still with Philips Research, and then that's when I felt that I really wanted to do something very applied, and that's when I was always in the industry. So I've always used to think that we don't diagnose enough. The devil is really in detail. The moment you see cancer, first thing we do is remove, get it out of the body, then do some biomarkers based on tumors and start treatment. So we need to really understand more of the tumor. Only then you can come up with either understand the tumor biology and come up with better biomarkers to make targeted drugs. So that is what I wanted to do, was develop a prognostic test. So as a scientist, I didn't know anything. I just had that idea, but how do I execute it? And that's when entrepreneurship is a very lonely road. I mean, you really don't have anybody. If you don't have a co-founder, you have nobody. Because where do you, so I was just talking to my husband, okay, I've resigned from Philips and I want to do this. And then, so even to incorporate company, you don't know anything. There's no guide. So you go to some of these, people who incorporate companies, lawyers and all that. And then they said, okay, yeah, you come, we'll do this paperwork. Then getting a name for the company, MCA decides what name you should give. They gave me, okay, give me 5 options, and they chose something else. I said, that's not that's my 5th option. Give me my first option. They say onco is very common. I said, yeah, because cancer is common. So, so your struggles really start from even getting a name for your own company. But all of them are very solvable. I mean, there is nothing that is this thing. You just need to be persistent. So incorporate the company, then who will fund you? There are no grants. I mean, there are grants, but at that point, you have no data, right? So who would give you grant on what basis? And by the time the grant cycles comes and they review and they give you money, time has gone. So I didn't want to approach that, because if I didn't do it at that time, I would have never done it. So I approached venture capitalists. Even there, some friends who from IISc who have done this, they say, oh, Manju, this is the worst time to raise money. 2008 bust has happened. It will be difficult. I said, when is it ever easy to do anything in life? There is nothing easy. I mean, crossing road in Bangalore is also hard. So I said, fine, let me just give it a try. And I approached, this is all where luck matters. So I became member of what is called the Indus Entrepreneurs. It's a TIE. It's a association of all entrepreneurs, and then one of them said, "Oh, you have recently become a member. Come and get mentored." I said, "When?" She said, "Yeah, tomorrow evening." I said, "Okay, I'm not really ready. I had not yet made any pitch deck or anything." Then she said, "Oh, don't worry, just make a business model and come." I said, "I don't even have a business model in mind. All I know is what I want to do." She said, "Never mind, just put down what you want to do and come, and how will you monetize it?" So I said, "Fine." And then I said, "Who are the mentors?" And the mentors were from nobody from life science. One was from Tommy Hilfiger, and one was somebody else CEO. So very different, areas where, and they were the mentors for that day. So I said, "Okay, I mean, what's there to lose? It's in the evening. I don't miss a work day. I can make a PPT." So I went, and my investors were in that meeting. And they said, "Oh, we like your pitch, and we support companies at this stage when they only have a PowerPoint." So that's what, don't miss our opportunities to interact with people if you want to ever be an entrepreneur, because that is where all the connections are made. And that's where I met them, and they funded me 4 months later after asking me questions after question. How will I do this? How will I do this? They understand the importance, understand the value. How will I do? Do I have a thought process in mind or not? So a lot of questions, but very important question. They also make you think, they make you focus in a direction that you really want to go. And then they finally say, "Yeah, fine, we'll fund you. Where is your lab?" So I said, "Yeah, I'm looking for lab." So I had nothing, right? I had no money, I had no lab, I had no tissue samples, and I had no people. So that's why I say, you're very lonely in this journey. So they said, "Okay, so you have any options?" I said, "No options, because in Bangalore we have 100 tech parks, but they all give you 10,000 square feet of space, and I didn't need so much. I don't have money to pay rent for so much." So I said, "No, I don't need so much space." So they say, "We have seen you struggle for space. Just have your office lab in my office. We have enough and more space." I said, "Okay." So I remodeled their part of one big room they had, and that was my first lab. So that's again, where luck really plays a part. And first hospital who gave us actually samples was Manipal Hospital, Bangalore, and also Hinduja in Mumbai. And the way I met Dr. In Manipal was, again, I was with my sister in some exhibition, and she kept nudging me and said, "Hey, this Dr. Poonam is right next to me." I said, "Really? Okay, let's just get introduced right away." So I got introduced to that doctor in that exhibition, and then I told her, "I'll come and meet you in Manipal tomorrow." And she liked the study. She said, "Yeah, let's do the study." So this is once again, how luck really plays, because working with clinicians is not, I mean, you all know this, it's not really the easiest, because they are busy, they are different, their priority is not to work with entrepreneurs, their priority is to treat and ensure that the patients do well. So they really, and they're inundated with patients. They don't get a single day to do any research studies, even look at their own patient data. And they've told me this themselves. So it's very hard for them to work. So you really have to meet them times when I have never met them with 1 hour to spare, just like 10 minutes. So you need to be so focused and so to be precise that this is what I want to do. And are you willing? And whom all do I need to meet? So, and then next day I go and I meet this guy, that guy, and then by the time you align all the ducks, it takes 5 months, and you visit 5 times. But eventually it gets done. And so I must have visited about 70, 80 hospitals in India alone, and we worked with 12. So that's the ratio you typically have to visit to really meet so many this thing. And our first task was just create data. So we needed to get tumor tissues with 5-year follow-up, and that is a huge challenge in India. Nobody keeps follow-ups. Nobody keeps patients' files. Some of them keep patients' file. Some of them give it away to the patients. Some of them give tumors to the patient. So I have heard all kinds of combinations and permutations. And I'm like, how do you guys seriously, do you understand the value of this? Some of them scan them very thoughtfully, but they never update the systems. So somebody who scanned in 2008 with Wipro, now they have Cisco. So now they can't open any of those files. So I have gone through all of these issues to really get to the data, and we needed 5-year follow-up, and I'm very strict about that. So I told him, if you don't have 5-year follow-up, I can't work with you. So only give me patients for that year that you have 5-year follow-up. So that's how it takes. But when on the other side with entrepreneurs, with investors, they say, where is your patent? So we needed to file a patent. So we needed to create data to file a patent. You can't just file from thin air what idea I had. So that really it was just intense pressure to really get data, file the patent, and then continue the data generation drive with just 3 of us. We were just 3 of us who would go to different cities in combinations or alone and speak with doctors, get the agreement signed. And there are many doctors who have asked me, okay, next time you come with a man and we'll sign the NDA. And to your point, Sahar. So I said, this is my 3rd time I've come. And we have discussed this, you agree. Everybody agrees. And there is really no man that can come and sign this. So why delay it to next time? Why don't we just do it now? So then they say, oh, okay, so you can sign. I say, yes. So then they would sign the agreement with me. So it really takes every ounce of patience and perseverance in you to do something that, and unless you have passion, you wouldn't do this. So finding statistician, there are no statisticians. I went to ISI Bangalore, I went to ISI Delhi, and everybody told me, yeah, this is very important. But you just tell me how to do and we'll develop a model. So I said, I have my degree in cell biology. I don't know nothing about statistics. If I knew, I would do this also myself. So I cannot guide you on how to develop that model. You ought to know it, or I find somebody else. And so I contacted all my friends back in the US and I said, hey, send me statistician names. And I contacted them, I went and met them, and out of trying 5 statisticians, finally I decided to work with 2. And actually one of my statisticians who had colon cancer died due to chemotherapy. He developed rapid leukemia, and he passed away during our work. And it was really, I still, there's some people you are so, I met him maybe once, but you connect with them so well. And I was really sad when I learned about his rapid leukemia that he developed as a side effect of colon cancer, which had recurred after close to 15 years. And he passed away due to that. So, but the other statistician who is also in the US is Ukrainian. He really worked hard to get this. They really have the flavor to do this. They know how to develop this, which would stand FDA certification tomorrow. And so they started work on 2014, and we finished it about middle of, almost end of 2015. 2016, we validated Canvas's breast, and we got all the certifications, submitted publication. Publication was another big uphill task. If you are, you guys know this, if you are not in academic institutions or in hospitals in India, it is pretty much impossible to publish. If you are a pharma, yes. If you are a startup, which nobody has heard of, good luck. So we tried many different ways to publish this in different journals, combinations of data, and finally, it just takes time. So finally, first publication really came out in, I think, 2018. Yeah, CAP paper. So 2017 end or 2018, I forget. So it was really, and since then we have been prolific in publishing, but it really was again a huge struggle to get that publication. And then you need to do validations in different countries. And my investors, why do you want to do it in the US? Why do you want to do it in Europe? I said, tomorrow if I want to sell in the US and Europe, who would buy this if there is no validation on their patients? So then they say, why you have in India? I said, no, but you need to do from their patients. I want to understand the ethnical differences, if any. So we found a very cost-effective ways to do this, because we also did not have enormous amount of money that we could sponsor these kind of trials in every country that I wanted to. And finding collaborators who would work with an unknown company, an unknown person in India. So that was really the hardest task. But in none of these hospitals I have mentioned, I have gone more than once to get them to sign. It was all discussions before, calls before, and then just one visit, and then they would sign the agreement. And they're so methodical about this. If I tell them I want to do a 500 patient study, they'll say, wait, let me first make sure we have 500 patients with this category and the follow-ups and the tissues, and then we'll agree to the numbers. Whereas here, I've seen so many hospitals, oh, 500, nothing, we'll give you 1,000 tissues. And then they gave me 50. Which is so frustrating because they don't have the tissues, no follow-ups, no proper follow-ups. Sometimes instead of breast, I get ovarian, or instead of breast, I get uterine. So, if I didn't have great pathologies, I wouldn't be here, talking about this. So these are all the things that you really have to be prepared to really achieve what you want to achieve. And then, of course, getting accepted in conferences is also not easy. The same abstract, if I submit, they may not accept. But a hospital submits, a collaborator submits, they would accept. And this has happened so many times. So nowadays I say, yeah, please submit. And you present. I have no eagerness to go present in any conference either. I will come there, but you present the poster or you present the talk, because anyway, it's because of you that we are even presenting. So we publish in ESMO, ASCO, I mean, all the COBRA, they've called us for presentations, but it takes time. And then all of these countries, every time I'll go for a conference, I'll visit other countries or meet those guys in the conference and completed all of these trials. And now our final validation is close to 4,000 plus. And then, of course, now you may have a fantastic test, but who's going to sell it? Selling is the most difficult task that I have done ever in my life. Developing CanAssist was the easiest task and the most, really satisfying. I guess maybe that's the background I come from. But selling it, you may have a fantastic test, but you still need to have a customer who would buy it. And that customer in my case are two. I have a patient who will pay and a doctor who would prescribe. So you need to convince both these guys, and then they would prescribe the test, and patient should buy it. So it's doubly difficult. And one of the mistakes I did was we didn't really publicize so much about, oh, we are doing this, we are doing this. We thought, okay, let's just develop this and then we'll see. I didn't realize how important it is to market yourself. And so we developed, and then in Bangalore, people knew, so they were using it. And then somebody I met in some conference said, oh, we didn't even know about this. Why don't you come and give a talk? So I said, okay, I'll come and give a talk. So that's when we realized importance of really giving talks in Trichy Oncology Consortium or Delhi Oncology Group or Mumbai Oncology Group, etc. So that's when we started actually giving talks about what we have developed, and then everywhere it takes time, right? They say, oh, run our samples, then we'll be convinced. Fine, send me your samples. So that's when it started, and then we didn't have sales members. And sales teams are expensive because whether or not they sell, you have to pay their salaries. So, and where are the revenues, to pay all of these salaries? So it's really like a chicken and an egg. But unless you have salespeople, it is a very high touch point. The patient is not going to go to the hospital and send you the tissue. You have to help the patient because patient is sick. So the sales guy needs to go to the hospital, take an authorization letter, get the tissue and ship it to us. So it really requires a lot of coordination. We did that for 2-3 years. And then last year we decided we are not good at selling. So let's accept. Let's work with somebody who's good at selling. And that is Zydus. So pharmaceutical company. So now we have tied up exclusive India-wide sales and marketing with Zydus. So they would bring in the tissue, but Oncostem will still run the test and report it. But entire coordination across India of picking up tumor tissues is done by them. And we would still go and give talks that they would arrange in every hospital, cities, conferences, wherever it may be. Oncostem will give the talks, but they would help to organize because they know clinician, they know how to run this. They are pros in that. So why invent the wheel? So we started working with them, and yeah, so let's hope for the better future and we improve our sales. So I wouldn't have done this without, so totally Oncostem's people are about 25 today. And we have always been around this range. We have never exceeded 25 to 30. With the sales team, we were about 32. And these are really, without these people, I wouldn't have been here. She's a very talented pathologist. Aparna is a researcher and operations and finance. And so how does one achieve? I mean, you have heard me say all of this, but I mean, for all the young students here, really, the one thing that you must do is be organized, manage your time, and listen more than just speaking. So really, that is one important thing I've seen, even in amongst my employees. I see sometimes they lack. And in case of business like ours, you really need to know your customers. I mean, KYC is really important. Because even if you have the best of your product, does your customer want it? So you should know, because there's no point developing a fantastic test, whether cheap or expensive. If nobody wants it, then you wasted your time and money in just developing it. So I mean, this is another thing I've learned. So many hospitals I've worked with, they didn't work out, but that's okay. You just leave and let go and move on. But be persistent in the hospitals that are positive. Work hard. This is one thing taught to me in the US. Work hard, but work smart. So you know where to stop, where not to go forward. Think differently. So when it came to prospective trials, everybody said do randomized prospective trials. Those take enormous amount of time. So today you recruit patients that will take you 5 years to recruit 500 early stage hormone positive, HER2 negative patients who are willing to be part of your trial. and then you follow them for 10 years, and then you analyze. So you're looking straight at least 15 to 17 years. Are my investors going to wait for that long? No. Will I survive without sales? No. So you need to think differently. So I said, yeah, I'm not going to run a trial, but I'm getting samples from studies like this, which are already completed elsewhere, and I get part of them in a blinded manner, and I run those in canvases based on those samples and show you the same data. So then they are agreed, and we already finished one Dutch trial, team trial, and the Dutch team was so excited by that data, they said, we'll give you another trial called Ideal. So that's where, think differently, dream big, and teamwork. I wouldn't have done this. I mean, there are just so many new things I learned about incorporating a company in India versus in Mauritius versus in Singapore, all the cross-border taxation, where to incorporate money, where to file your patent. All of this wouldn't have been possible if I didn't have the right finance guy, work with all these big fours, EY, or whoever it is, to understand the cross-border taxation, to realize what makes economical sense 10 years later, not today. So that was another, because they always think when you go public, when you are sold, having an incorporation in India is just going to work against you. I said, oh, you think so far ahead. You say, yes, you have to. Because otherwise you will, unless the exit is there for a company, nobody would invest in the company. So there are all these things that I really learned along the way. I have absolutely zero regrets, because it's really very nice to know different things and something different that I've always enjoyed learning. And finally, it's your mindset. And I want to really end with this mindset video. Teamwork, I think all of you know, so I won't show the mindset teamwork video in the interest of time. So let's just move on to, it's all about the mindset. So this is a video about this breaking a barrier for running 1 mile, which is 1.6 kilometers under 4 minutes. And until 1886, nobody had done that. I mean, they were all very close. Until 1954, they were all very close. You can see the timelines: 4 minutes, 6 seconds, 4 minutes, 1 second, etc. But nobody could do it below 4 minutes. The best of athletes in US and Europe could not do this. So everybody kept thinking, is it perfect conditions, wind resistance, my mood, my carb loading in the night before, etc. Or it was more of a psychological barrier than a physical barrier. Literally, that was the situation. But then this doctor did. Roger Bannister was a doctor himself in Boston, and he really conditioned himself to do this challenge. And he did it in a wet day, absolutely non-ideal conditions on a wet track. He did it under 4 minutes. And since then, there are many athletes who have done it. So it was just the mindset that changed when he did it. And that's the video I really want to show you. Who was that who said he is? It's in the, here, this one?
Yeah.
Okay, so okay, here, yeah.
You want to tell me that circumstance matters more than mindset. That may be your perspective.
Oops. Oh. Okay. I can't do the full. Okay, yeah.
You want to tell me that circumstance matters more than mindset. That may be your perspective. But I'm telling you, I've seen the opposite. I've seen mindset change everything. I've seen teams go from being broken to winning championships. Same team, same talent, different mindset. Think about this. In May 1954, Roger Bannister broke the 4-minute mile. And in the sports world, it was a huge milestone. Here's the backstory. Go back to 1942. During World War II, the record for running the mile stood at 4 minutes and 6 seconds. Then over the next 3 years, during the war, the record got whittled down to 4:01. That's where it stayed for 9 years. No one could break 4 minutes. Performance in that distance was stuck. Then, through incredible teamwork, Bannister breaks 4 minutes. Did it by sticking to the pace of one teammate for the first 2 legs.
My legs seemed to meet no resistance at all. almost as if impelled by an unknown force. We seemed to be going so slowly.
And then another teammate for the third leg.
I was barely aware of the fact that Chris Chataway was now going into the lead.
Before going on his own and breaking the record in the final leg. It was an incredible achievement.
A 4-minute mile was possible. Somehow, to do it, I had to run the last lap in 59 seconds. Chataway led round the next bend, and then I pounced past him at the beginning of the back straight, 300 yards from the finish. I had a moment of mixed joy and anguish when my mind took over. It raced well ahead of my body and drew me compellingly forward. I felt that the moment of a lifetime had come. The faint line of the finishing tape stood ahead as a haven of peace after the struggle. I leapt at the tape, like a man taking his last spring to save himself from the chasm that threatens to engulf him. My effort was over, and I collapsed almost unconscious with an arm on either side of me.
Here's the crazy part. A month later, the first woman, Diane Leather, breaks 5 minutes. A few weeks after that, the second guy, John Landy, runs under 4 minutes as well.
We have unofficially here 3:58.9.
And within a year of Roger doing it the first time, 5 men had run the mile in under 4 minutes. So what changed? Their mindset. Now that Roger had reached the mark, everyone else knew it was possible. Impossible became possible. Because it's always harder to raise the bar than to catch up to someone else. And the limitation is your mindset. So what is your team's 4-minute barrier? What is the limitation that makes you feel like you're stuck? Ask the question. How are you getting in your own way?
Thank you all for listening. We'll be happy to take questions.
Thank you, ma'am. Sure. Yeah. May I now request Director Sir to kindly felicitate ma'am.
That's for everything.
Sit, I think they'll.
You can ask some questions.
Then we can bring the chairs.
Two chairs.
Have any questions?
Hello.
Yeah.
Very nice talk, Dr. Bradley. So I have a small question. The CAN assist that you have mentioned. So you have a score, risk score, which you said that 15.5 is the score, that most of the 70% of the affected those who are undergoing the test, they fall under that, and then rest 30% falls under above 15.6. But you have